From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 23, recorded on October 25th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York, Tim Chung.
Hi, Vincent. Hi, everyone. Nice to be here.
Welcome back. It's kind of gray out in New York today, isn't it? Huh?
Yes. And it'll be pouring down soon, apparently.
Yeah, we're supposed to get a lot of rain, right?
Yes. Flash flood warning. So,
also joining us from Nashville, Tennessee, Vivian Morrison. Welcome.
Thanks. Hi, guys.
Are you expecting a big storm?
No? I don't think so. I mean, it rained last night.
Right.
It's moving up.
Okay. All right. And we have a guest today also from Nashville, Tennessee, Bruce Carter. Welcome to TWiN.
Hi, everybody. Great to be here.
Hello.
And just full disclosure, Bruce is Vivian's postdoctoral advisor, right, Vivian?
That's correct.
And so it's great to have you, Bruce. And we're going to start off by hearing a bit of your background, where you're from, where you were educated, and so forth.
Sure. Well, I grew up in Michigan, and I went to a small liberal arts college for my undergrad. School's called Alma College. And then from there, I went on to the University of Michigan to do my PhD. And that time I was doing research on opioid receptors. And we still didn't know a lot about the signaling mechanisms there. So I defined how the opioid receptors worked with certain G proteins. That was my thesis project. But in the process, I decided that I wanted to move on to something different. And I met this guy, Hans Thunen, at a meeting and talked to him about neurotrophins. This is a family of growth factors, basically, that promote the survival and differentiation of neurons. And so after talking with Hans, he suggested that I might contact his collaborator, Eva Lombard, and talk to him about doing a postdoc with him. And so I did. I contacted Eve, and we exchanged emails and phone calls. And then I eventually went over there and visited and decided to do a postdoc with him. So just a bit of background. Eve is the one that discovered brain-derived neurotrophic factor, or BDNF. Some people may have heard of that, given its role in synaptic function, especially. And so I went to work with Eve, and my project there, he got me interested in looking at this other neurotrophin receptor, not a classic tyrosine kinase receptor like most of the receptors we knew about. There was another receptor called P75, which was kind of a mystery. And so I started working on that. And that was over in Munich at the Max Planck Institute, which is where Eve was at the time. And so after 3 years in Germany, I really enjoyed my time there, but I thought I really needed to get back into the American system to look for a job. So I kind of continued on in the neurotrophin field and went to work with Moses Chow, who at that time was at Cornell Medical School, now called the Weill College of Medicine. And he was the one that actually first identified and cloned P75, this receptor that I was working on. So I worked with Moses for a while on that, and then started looking for a job and ended up getting an offer here at Vanderbilt and decided to take it, and I've been here ever since.
I know Moses. Moses was a postdoc with Richard Axel, right?
Yeah, exactly. I was telling Vivian recently that it was quite a crew that went through Richard Axel's lab at a time. Linda Buck, who shared the Nobel Prize with Richard Axel, was there. Another very well-known developmental neurobiologist David Anderson was there. David Julius, who just won the Nobel Prize, was there. So all these guys and ladies at the same time was a pretty remarkable crew.
Yeah, it's and also there's this little company up in Westchester called Regeneron that was big into BDNF, right? Didn't they make some product, or at least were testing it?
Yeah, so they're the head of that. I'm not sure if he's still, how much of it is involved. George Yankopoulos.
Oh yes, yeah.
He was an immunologist, but really interested in neurotrophins for potential therapeutic use for things like Parkinson's disease and other neurodegenerative conditions, given their known role in promoting neuron survival. And so, yeah, they were really into neurotrophins for a while. But, once we discovered how complex it is to treat these conditions and to use neurotrophins, they completely abandoned that project and don't do that kind of stuff anymore, as far as I know. Yeah,
George was a MD PhD at Columbia, actually. I remember when he was a student.
Oh, wow. Small world.
Yeah, now he's wealthier than all of us put together. It's amazing.
Yeah, for sure.
Wow.
I mean, probably not hard when you're talking about Tim and me.
Well, all of us.
I think my wealth is in the negatives.
So when you were an undergrad in at Michigan, were you already interested in a career in science when you came in?
Yeah, pretty much. I've always had an interest in, science. I mean, I kind of thought I might want to do the MD route, but I knew I wanted to do research. But pretty soon I discovered that, dealing with patients wasn't my thing, and I much preferred just being in the lab.
Yeah, that pretty much mirrors me, and I think Jason as well, one of our other co-hosts.
So Bruce, you're okay with Vivian doing This Week in Neuroscience, right?
Yeah, I think it's a fun thing, and I think it's pretty cool that you guys are doing that.
Yeah, we have a lot of fun here. Well, I'm going to hand it over to Vivian to kind of guide our discussion about the work in your lab.
Yeah. Thanks again, Bruce, for coming on such short notice, but I actually think it's a great opportunity to touch on a couple of things that I've mentioned in previous episodes, some kind of themes in neuroscience that I really enjoy and I think are useful for our listeners to also know about. And that is, first and foremost, that your nervous system is composed not just of neurons, it's composed of many different cell types. And it's not just your brain and your spinal cord. It also involves nerves, some of which you can consciously control, some of which you can't. And so we know kind of in a hand-wavy way about some of those, but I think it would be enriching for the TWiN listeners to hear a little bit more about the peripheral nervous system, so those nerves, some of which you can control and some of which you can't, and that the latter is the autonomic nervous system, which Bruce also has some expertise in. So I guess I want to just do a really, really brief introduction to the peripheral nervous system, and then Bruce can dive into a more detailed view of its development and function. Another theme here that I hope he touches on is one that's of interest to me personally. It's the overproduction of cells to make sure you have enough, so to speak, to do what you need to do. And then there's a orchestrated culling of the cells that are not required for the developing nervous system. So, and the P75 neurotrophic receptors involved in regulating that. So I'm sure he'll talk about that. But so basically, like the really basic breakdown of your nervous system is your central nervous system and peripheral nervous system. So the central contains your brain and spinal cord. But as soon as you exit the spinal cord, you enter the peripheral nervous system. And so those are all your nerves that are in your limbs, as well as those that innervate your organs, like your heart and your intestine and blood vessels. And the latter group of nerves are part of the autonomic nervous system. So they're doing all of these things that you don't have to think about, like regulating your heartbeat, regulating where blood goes in your body, whether you've just had a big meal and so blood is going to get shunted to your digestive tract, or if you're, to use the frequently used example of your fight or flight response being activated. If you're running from a lion, you need that blood not to go to your digestive tract, but instead to go to your muscles. So really important, and actually a really important thing for everyone to know about now, because as we probably all kind of know, that your autonomic nervous system is constantly being slammed by the stresses of modern life. It's not a lion anymore, it's something else. And so I think people are starting to be like, oh yeah, there's this other part of my nervous system that maybe I should try to do what I can to get in touch with. But in any case, the parts of the peripheral nervous system that you can control include your sensory, or your motor neurons, but your motor neurons are going to respond to things that come through the sensory neurons in your nerves. And so that's basically like the bare bones of it. And my point there is just to show everybody kind of how the peripheral nervous system is composed of these two different branches, autonomic nervous system and that which is not. Yes, Tim.
Sorry, I got a question, which is, what if I have a neuron in my spinal cord, like a motor neuron, whose cell body is in the spinal cord, but it sends an axon out of the spinal cord to my muscles? Does that count as part of the central nervous system or the peripheral nervous system?
That's a good one. Bruce, you want to field that one?
I would say both.
Okay, that's fine. I was just.
I mean, motor neurons are definitely part of the CNS, but because their axons project out to the periphery, they're like, for example, they're myelinated by Schwann cells, which are the peripheral myelinating cells. So they definitely have aspects to the peripheral nervous system to them, but they have more complex functions in the CNS. For example, they're influenced by microglia, which are only found in the CNS. And Vivian, I want to mention one other sort of nervous system that we do usually lump together with the peripheral nervous system.
I think I know which one this is going to be.
The enteric nervous system.
Yes. Bruce is all about that gut microbiome, all those, we have more dopaminergic neurons in it's serotonergic. Is that what it is? More serotonergic neurons in our gut than in our brain.
Yeah,
so, yes.
I know a number of colleagues that would really hate to be forgotten.
Would one of them be Michael Gershon?
Yes, definitely at Columbia.
Yeah. I've learned about the enteric nervous system from him. Yeah.
Do you want to expand on that one? Since I don't really know much about it.
Is it true? I heard this fact that there are as many neurons in a human's enteric nervous system as there are in the brains of a cat. Is that correct? Supposedly.
I don't know, but I know the diversity of neuron subtypes in the gut is arguably as much as in the brain, just because there are so many different transmitter types in the gut. But that's not my specialty.
Oh, okay. Well, we won't go that way.
Well, someday we need to get someone in who works on the enteric nervous system. That would be good, right?
Yeah.
Talk to Michael.
I think we've talked a little bit about that stuff in relation to like what the gut microbiome is spewing out and fatty acids and stuff, and how that had some impacts on central nervous system function.
Well, some of that actually is through the sympathetic system.
Mm-hmm.
Invades the gut and is responsive to the microbiome, and actually the microbiome is responsive to the sympathetics, which I think you brought up an interesting point about stress, Vivian, where that is that is the sympathetic nervous system being activated. And there's good and bad in that, right? Because in modern life, too much stress is bad. At least the high levels of cortisol has all these negative things associated with it. But on the other hand, like if you're giving a presentation or you're going into military, you want to be hyper aware. So if you're giving a presentation and you want to be like on your toes and remembering everything you can and talking to the audience and responsive, that sympathetic nervous system makes you hyper aware and makes you really in tune to all these different things. And so that's a benefit. So, there's good and bad, and the stress that can happen. Some of the bad things are, the known issues we have with stress over time, like on the cardiovascular system and actually on the gut microbiome. That's some more recent data about how sympathetic release of things like norepinephrine can affect the gut microbiome.
Speaking of that, can you talk a little bit about what we're starting to learn about the relationship between sympathetic neurons and tumor growth?
Yeah, so that I've learned a lot about that recently because I have a colleague here, Marcus Tan is his name. He's actually a surgeon who works on pancreatic cancer. And one of the things that he's interested in, and he's kind of roped me into the project as well, is the fact that the pancreatic cancer, which probably many of you know is one of the most deadly forms of cancer, that when the cancer grows, it releases factors like nerve growth factor that help more innervation happen. And that's a bad thing because as the innervation of the sympathetic nervous system in the tumor happens, then when the sympathetic nervous system is activated, it releases norepinephrine there. And the norepinephrine actually helps the tumor to grow. These tumors have the receptors on them. And so over the last few years, we've learned that that's one of the key ways that stress is actually a bad thing in cancer, because as you get more stressed, your sympathetic nervous system is activated, and that causes your tumor to actually grow. And it's turning out to be true in a number of tumors. So the same has been found for prostate, for example, and I'm pretty sure lung as well. So yeah, it's another example of where stress can be a negative thing. And we don't understand all the details of the mechanisms involved there, but that's one of the things that Marcus is interested in exploring.
I don't know if Marcus has thought about this, but I was just wondering if, how sometimes people can have prescriptions for medical marijuana or can do basically regulated or controlled trips on psychedelics. Do you think that those in any way would probably be targeting sympathetic nervous system? I mean, it's not like you're going to do, whatever it is, psilocybin all the time while you're, doing your cancer treatments, but you might be consuming cannabis in some way. Is there any known relationship between decreasing sympathetic nervous system activity and cannabis and the cannabinoids?
I'm not familiar with that. I don't know if the sympathetic neurons have the CBD or the cannabinoid receptors. I can imagine, though, that top-down mechanism where, the cannabinoids have an effect on the CNS, and the CNS definitely affects sympathetic tone. So it may be that you feel more relaxed and less anxious, and that reduces the sympathetic and maybe activate some of the parasympathetic, which, we're talking a lot about the fight and flight response. We have an opposite side to that, where our parasympathetic nervous system is more of the relaxation type of response. So, when you're calm and that your heartbeat slows down, that's the parasympathetic is helping to do that.
The mnemonic that I learned was rest and digest for the parasympathetic.
Exactly.
Yeah. I remember in my first year, the first year curriculum for the my graduate program in neuroscience at Georgetown, one of the test questions on when we were going through this module about autonomic nervous system function was, which of the two branches is absolutely necessary? They're like, you couldn't live without either parasympathetic or sympathetic. And all of us were like, oh, sympathetic, like you definitely need sympathetic nervous system. The rest of the other stuff's like not as important, but it's actually the reverse. If you don't have a parasympathetic, you can survive with, I mean, probably not well without your sympathetic nervous system operating at full steam, but that parasympathetic nervous system is really important for like tissue and organ homeostasis over the long run. Because if you don't have, for example, enough parasympathetic innervation or activity in the heart, then that can lead to heart failure later on. And I'm sure there's other examples in other tissues, but I just thought that was funny. It was the complete opposite of what the rest of us had thought.
Yeah, well, that heart regulation, that's a key sympathetic and parasympathetic target. I mean, there's a lot of complexities there, different parts of the heart being regulated by sympathetic and parasympathetic input, and can affect everything from heart rate. When you're nervous, your heart rate goes up, obviously. Vascular tone is controlled by the sympathetic system. So there's a lot going on there.
Mm-hmm. I was wondering if we could switch gears a little bit and talk about your work with P75 and that theme I mentioned earlier of having an overproduction of neurons and then kind of scaling it back and selecting which ones survive and which ones don't. And I think it might also be cool for you to introduce to us the kind of combinatorial function of P75 with its receptors, because I think that's another really cool theme in biology. And we're learning more and more in neuroscience that it all depends on who's present and with whom and when. So I was wondering if you could just take us through this kind of developmental cell death among sensory neurons and then how P75 plays a role in that.
Sure. So as you mentioned, normally we have about twice as many neurons generated as we need. And that's true in the periphery and in the CNS as well, although in the CNS, it's a little bit more heterogeneous. Some populations you get twice as many, some populations there's about 10% cell death, some more. So it varies a little bit in the brain. The question?
All right, I do have a question, which is the overabundance of neuron is usually done during development, right? When we're either when we're still an embryo or early postnatal, I don't know when, but like when we're very, very young. But I'm wondering, I normally thought when we're young, our brain is very small and our spinal cord is very small. So it's kind of weird that there are a lot of neurons there. So how come as we get older, our brain gets bigger, but there are fewer neurons? Where did all the space go? I'm just wondering.
I would say that there's probably a couple different answers to that. One is that, you have a really dense number of neurons to begin with, but then they start putting out their branches. And so you get this very complex arborization of all the synapses being formed.
Okay.
And then the other thing is, of course, like Vivian mentioned, there's this other important cell type called glia that the neurons are generated first, and then the glia populate the brain and in the peripheral nervous system too. And they are really important in regulating things. So they're kind of taking up some of that space as well.
Oh, okay, great. Thank you.
And the cell death, actually, it's an interesting theoretical question. We often say, well, the cell death is a way for complex organisms to be able to be plastic in their evolution, so that certain neurons are going to die because they're not being used as much, or in that organism are not that important. And so this is a way to refine the nervous system. That sounds great, but that doesn't really fit with a lot that we know. Just as an example, in the brain, it's definitely way more complicated than the brain. There was an example a few years, well, probably like 10 years ago now. I think it was Arturo Alvarez-Bulla, but I'm not positive on that. They did a study where they injected these neural precursors into the brain in different amounts, and then looked how many survived. So if there was some limiting factor or, the classic half of them are going to die, you would expect that no matter how many you inject, half of them will die. Well, that didn't really fit with what was observed, that the number of cells that survived was really not dependent just on how many there were. And the other thing that's weird is you see cell death in C. elegans as well. And they've mapped out C. elegans, like every single neuron that's there. And it's always the same one that dies. So if they have like these two neurons right next to each other, it's always this one that dies. So that idea of the plasticity doesn't quite seem to fit with a lot of the data. But I don't have a great answer for it. It's an interesting conundrum. And in the peripheral nervous system, it's definitely 50% of sympathetics, 50% of sensory neurons die. And that is regulated by these factors called neurotrophins. So, for example, your heart, we talked about innervation of the heart. The sympathetics will innervate the heart, and the heart is producing nerve growth factor. And so the sympathetic neurons need nerve growth factor to survive. And actually, it was, we were talking earlier about some of the Nobel Prizes. One of the Nobel Prizes went to Stanley Cohen and Rita Levi-Montalcini for the discovery of nerve growth factor. It was the first growth factor that had been identified. And they showed that it promoted survival of sympathetic and sensory neurons. And if you give more nerve growth factor, you get more neurons surviving. And if you, like the classic experiment they did was with a chicken, cut off one of the limb buds. So there was this less target tissue. And so then they saw more neuron cell death. So the amount of nerve growth factor in that target tissue is really important for regulating survival. But then, as Vivian said, and we started looking at how this works, we've discovered that it's actually pretty complicated. So these neurotrophins, like nerve growth factor, and I earlier mentioned BDNF, they act through a family of tyrosine kinase receptors called the TRACs. And they activate typical growth factor signaling pathways, and that promotes survival. And so the idea for a long time was that, you need the TRAC activation to get this survival. And so if you don't get enough of that signal, then the neuron dies. Well, it's not exactly how it works, though. So it turns out that the nervous system and some of these targets actually produce signals that actively kill the neurons. And so there's this receptor, which was actually the first neurotrophin receptor identified, called P75. So back in the early days, like 1970s and 80s, people were looking for the nerve growth factor receptor. And the most prevalent thing you could find that binds nerve growth factor is this P75 protein. So that was cloned by two groups, one being my former mentor, Moses Chow, and the other being Eric Shooter, who was at Stanford. And so they both had identified this P75 protein. The problem was that P75's expression didn't really fit with what was known about NGF promoting survival. It was on some neurons, not others, but it was on lots of other neurons and lots of other tissues. And so it didn't really seem to fit. And this P75 thing doesn't have any catalytic domain. So it's not a tyrosine kinase receptor. It's not a G protein coupled receptor. So it was kind of an enigma. How does this thing work? And then, so that was, the cloning was done, I think it was right around 89, 90. And then shortly thereafter, mostly through the work of David Kaplan, it was realized that there is a family of these proto-oncogenes called TRACs, and that the TRACs were the key survival tyrosine kinase receptors. So then for a long time, we were like, what the heck does P75 do? Well, the first thing we realized is that it actually forms a high affinity complex with TRACs. So in order for nerve growth factor, for example, to have its maximum survival effect, you need P75 and TRAC-A. And they form this complex, and it's high affinity, and the neuron's happy. But it turns out P75 actually has this evil side to it. So it's kind of Dr. Jekyll and Mr. Hyde. And so P75, when it's activated on its own, can actually cause cell death. And so, for example, in the sympathetic nervous system, we now know there's a critical balance between the survival signaling by P75 and TRAC-A together versus death signaling by, and that's what gives you the right number of neurons during development.
Can I ask
Sorry,
you?
Bruce,
sorry,
you cut off for a second. We didn't quite hear what is the second thing that was required. Could you repeat?
That sentence?
Yes, that sentence just now. Sorry.
Well, so when sympathetic nervous system is developing, you get survival signals through P75 and TRAC-A working together to give you survival. But you also get cell death signaling through P75 alone. So you get a critical balance of survival and death signaling during development. And you need both of those in order to get the right number of neurons.
Is P75 on the entire neuron and axon, et cetera, or is it just in the cell body?
That's a really good question. And right now, everything that we've looked at, it is all over the entire neuron. But one of the things that we've been studying more recently is looking at activation of P75 on the axon terminal. So, like I mentioned, these neurons are innervating their target tissue, and the target's producing nerve growth factor. Well, sometimes the target's producing other neurotrophins that don't activate a particular track. And so, in other words, not a survival signal. And so people have studied this retrograde survival signal for a long time. But one of the things we found more recently is you can activate P75 and actually get a retrograde death signal.
So this all sounds like, oh, go ahead, Tim.
Sorry, one more question. You mentioned P75 and TREkker can both bind to nerve growth factor. Is that correct?
Right.
And then when they do it in concert, it gives the grow signal, whereas if it's just P75, it is a death signal. So do they bind to the same region of the nerve growth factor, or do they compete with each other?
So they're binding to different regions on the trophic factor, on nerve growth factor.
Okay.
But it's not really a competition. So when that high affinity complex forms, and I should say there's still a lot we don't know about the molecular composition of it. From a lot of classic biochemistry, it looks like a dimer of dimers. So two P75, two TRACs, and they can bind nerve growth factor at the same time and give that high affinity complex. But some of the crystal structure analysis has been conflicting with that. And so that's why I say we still don't really understand exactly the structure of it.
And that's crazy to me, because you said, this people have been studying this for decades, and yet there's still not consensus about some of these things that should be really fundamental and basic. And you feel like they should already have been sorted out.
For sure.
And I was going to say also, the way you're telling the story right now is it's just the neuron, and it's just the neuron getting its survive or not survive signal. But a recent graduate from your lab actually did some work suggesting that the Schwann cells, which are in the same vicinity and also express P75, actually may play I don't know if you would say this, but the larger role in determining whether the cell dies or not. But as I mentioned to the other co-host, this is, there's a way of thinking that has been in play for many, many years. The neurotrophic theory of cell death. And it's hard to go up against that and say, like, how do some recent findings align with that, or how do they challenge it? I wondered if you could talk about how Schwann's, what she found, Rose Follis, what she found, and what it says about Schwann cells. Maybe we can talk about Schwann cells for a little bit after this.
Sure. I'd be happy to. So, yeah, as I said, this high affinity complex of P75 with TrkA, the assumption for 30 years was that is critical for giving this high affinity binding site and maximum survival signaling. And so if you got rid of P75, then you would have less of that high affinity survival signal. And so you would have more neurons dying. So if you look in the global knockout of P75, back when Quofen Lee did it with Rudy Janisch, was one of the pioneers of doing mouse genetics. They've knocked out P75 as one of actually the first genes knocked out. It was a Cell paper in 1992. And they noticed that the sensory neurons were lost more than normal. So you actually, with P75 gone, about half of the sensory neurons are gone. Even more than normally you lose half, well, you lose another half because you don't have P75. And so the assumption's always been that it's because the neurons don't have that high affinity maximum survival signal. Well, we were interested in looking at the role of P75 in those sensory neurons and really mostly in the Schwann cells. So P75 is also in Schwann cells. And just a reminder, that's the myelinating cell that wraps around these sensory neurons and motor neurons. So the full knockout of P75, not only do you have less sensory neurons, but you also have this hypomyelination. There's less myelin. And we didn't understand how that works, whether it's because P75 is gone from the neuron or gone from the Schwann cell.
And Bruce, just to clarify, it's not that, like, you might be like, well, of course that makes sense that there would be hypomyelination. There's fewer axons to be myelinated, but it's not that, right? It's that, like, if you looked at the proportion of axons that are remaining that are myelinated, it's lower, or that, and or there are changes with, like, the myelin thickness.
Yeah, it's both of those things. Yeah, that's thinner myelin and fewer myelinated axons.
Okay, so it's not just a function of there being fewer neurons. There's actually something specific either happening to the Schwann cell or maybe communication between the axon and the Schwann cell.
Yeah, exactly. Yeah.
Okay.
So we made a conditional knockout where we deleted P75 in the neuron and then also separately in the Schwann cells. And we had two really big surprises. The first surprise was that myelin was absolutely fine in both of these models. So we still don't know what the heck is going on, because if you have a look at the global knockout, you have this myelin problem. But if you knock out P75 in the neuron or the Schwann cell, the myelin looks fine. But the other big surprise. Yeah, go ahead. Do you have a question?
Sorry, I'm just wondering. So this is perhaps a little bit technical, but I'm guessing the knockout of P77 was
P75. I mean, it could be any. Just throw together some digits.
P75. I'm guessing that knockout was restricted to either neuron or to Schwann cell by, like, a Cree driver line. And Cree driver line, sometimes the Cree doesn't get turned on until different stages during development. Could that have something to do with why you have potentially different phenotypes?
Yeah, it could, but it doesn't seem to explain the myelin phenotype, or at least that doesn't seem likely. So the driver we used for the Schwann cells was desert hedgehog, and that turns on around E11, E12, which is like way before Schwann cells commit to being myelinating. So it's well before myelin starts. And in the neurons, we used a drawing a blank right now. I think it was the TUJ, the tubulin, alpha tubulin or beta tubulin driver. And so that would delete it in all the neurons, actually in the CNS and the peripheral nervous system. And so that comes on right around, kind of about the same time, actually around E12 or so. And that would be at a time when neurons are committing to being a neuron phenotype, a neuron fate, and as opposed to a precursor. So it's pretty early in development. But yeah, the myelin is totally fine in both of the knockouts.
And myelination, at least in rodents, happens almost entirely postnatally, right? I mean, of the peripheral nervous system.
Yeah, so it starts right around birth and then is done by 2 weeks.
And are there reductions in Schwann cell numbers in any of these mice, like the global knockouts or your 2 conditional knockouts?
Well, the conditional knockouts we didn't quantify because everything looks fine.
Okay. Yeah. So right. If it was an oligodendrocyte, you would definitely, you definitely have to look at that, because the oligodendrocyte, in contrast to the Schwann cell, has, I think the number, and I don't know, I looked for the paper once that purportedly said this, but it can myelinate up to 40 different axons. So it's very promiscuous. But the Schwann cell is like, I give myself to you, it, will only wrap 1 axon. But the axon obviously has many other Schwann cells on it, so it's not faithful at all. But so that you could see why I would ask that question, because there are examples of where if you change the number of axons or change the number of oligodendrocytes, then you can have like strange changes in like how many axons are myelinated and how many branches there are per oligodendrocyte and stuff like that. So it's my central nervous system glia scientist asking there.
Well, so the answer in the P75 global knockout, I don't think we have a really clean answer, because there are fewer myelinated axons, there are fewer axons, so of course there's going to be fewer Schwann cells. But I don't think anybody has looked carefully early on to see if the proliferation is affected at all. But in any case, that was the first surprise. And the other thing that was probably even more surprising was when we did the neuron knockout of P75, the number of neurons was not changed. But if we knocked out P75 in the Schwann cell, then we lost about 30% of those sensory neurons.
That always blows my mind.
Yeah, so this went against like 30 years of dogma that you need that P75 in the neuron to get maximum survival signal. And so we're saying, no, actually, it doesn't seem to make that big a difference. But if you knock it out in the Schwann cell, then you're losing neurons. And then Rose went on to characterize what was going on. So it turns out P75 has a role in lipid metabolism in the Schwann cell. And even though the Schwann cell can still do its job, it's producing this toxic lipid that seems to be killing the neurons. The Schwann cells are completely resistant to it, but if you add it to neurons, they die. I can tell you that the reviewers of this paper at multiple journals did not like that idea because, the dogma is P75 is important in the neuron for survival of the neuron. And, you can always make the argument, well, your Cree driver of deleting all of the P75 or from all of the neurons. And there's no perfect Cree driver that deletes every single, gene in every single cell it's supposed to. So, we cited the fact that actually another group had published a similar result where they had used a neurofilament driver. This was Simon Murray and his group in Australia. And they deleted P75 in neurons and did not see an effect on neuron survival either, or a very small effect. And they were similarly criticized. Well, neurofilament is known to not be a good Cree driver. It doesn't delete it very efficiently in a lot of the sensory neurons. So anyway, we eventually convinced some people that what we did is we actually took out the neuron knockout and just focused completely on the Schwann cell deletion. And then it got accepted at Journal of Neuroscience. So I still would argue, though, that you can do a deletion of P75 in the neurons, and it probably has some effect, but that's not the only thing causing the death of those neurons.
That's a call out to all scientists with resources to do this. Go out, knock it out in neurons, and get back to us. I was wondering if this might be a good time for you to, since we were talking a little bit about Schwann cells, and people who've listened to, if maybe they listened to the CNS Glia 101 episode we did a while back, they would be familiar with what myelin is. Just to recap, though, and I think Bruce mentioned it as well, that the oligodendrocyte in the central nervous system and Schwann cells in the peripheral nervous system create this insulation with their own membranes wrapped around the axon to maintain the integrity of the electrical signal that goes from the cell body down the terminal. I don't think I mentioned it before in that previous episode. We may have talked about multiple sclerosis, which is a disease in which the central nervous system, myelin, is attacked by the immune system. But I think there are a whole host of peripheral nervous system disorders where myelin, and the Schwann cells, something is wrong with them, and this leads to pretty debilitating diseases. And Bruce and some other people at Vanderbilt are working on understanding the development and progression and looking for treatments for a particularly devastating disease called CMT1A, or Charcot-Marie-Tooth disease. And there's some actually cool stuff that, Bruce, I don't know if you want to talk about it, concerning one of these myelin proteins, PMP22. That's somehow involved in CMT1A development, and kind of what that can tell us about myelination and myelin stability in the adult.
Sure. I guess one thing I would start with is, I think it was Vincent, you mentioned my fish. So I had a fish tank in the lab, and I used to have 3 fish, Charcot, Marie, and Tooth.
Wonderful.
So the disease is named after 2 French guys and a British guy. Jean-Marie Charcot was kind of considered the father of neurology. He described a lot of the classic diseases, including MS and important aspects of Parkinson's. And he then also described Charcot-Marie-Tooth disease. And his trainee was Marie, I can't remember his first name, Jean-Marie maybe. And then there was a British neurologist, Henry Tooth, that also described the disease at the same time. So my fish were named after them, and unfortunately the French guys died, but
this is Tooth?
British guys still alive.
Well, we got to get you some new fish.
Schwann cells are actually a topic of a lot of interest, at least in the glial biology world right now, because we're realizing more and more how much communication goes between those Schwann cells and the axons. Kind of the same thing that's being realized with the oligodendrocytes in the brain. So it's actually been known for a long time, probably at least 25 years, that if you get rid of the Schwann cells during development, the neurons all die, like the sensory neurons. So there's definitely something critical about the survival of the neurons. It depends on those Schwann cells. And there's lots of different signaling going back and forth between the Schwann cells and the axon. There's ideas about maybe metabolites helping the axon function. And if you think about it, it kind of makes sense. I mean, like your motor neuron that's myelinated and innervating your foot. The motor neurons in the spinal cord up there, like near your butt, and that's like a meter long in somebody who's around 6 foot tall. So that axon's a long way from its cell body. And so it makes sense that the Schwann cell is going to help out with some different aspects of its function. But we really understand very little of that. There's all kinds of weird things that have been recognized. So I remember there's one study where they had made ribosomes that were GFP labeled in Schwann cells specifically, and then they could detect them in the axons. So whole ribosomes are being donated from the Schwann cell to the axon. Now, functionally, we still don't know what that means, but anyway, Vivian brought up this disease, Charcot-Marie-Tooth, which it's really a spectrum of diseases that affect, it's all peripheral. There's no central aspect to the disease. And it affects peripheral neurons. And type 1 affects the Schwann cells. And so the Schwann cells have some problem, and then you undergo like a neuropathy. There's another kind called type 2 that seems to affect primarily the neuron, and they have a neuron degeneration that then results in a myelin problem. But the most so this is actually, Charcot-Marie-Tooth is the most common neuropathy there is. It affects 1 in 3,000 people.
So this is like on par with, I don't know about the numbers, but like when I talk to people about MS, I say, everybody knows somebody who has MS because it's so common. Can you say the same thing about CMT? Because I know that and we both know, like already there's like 2 or 3 people in my sphere who I know who have it.
I should say hereditary neuropathy. Diabetic neuropathy would be the most common form generally, but hereditary would be Charcot-Marie-Tooth. And yeah, I think the problem is that it's underdiagnosed. It's getting better, but it's long been underdiagnosed. That it's a spectrum. So some people have really severe forms and are in a wheelchair and never can walk. And then other people, it's fairly mild. They may have some muscle atrophy that limits their use of their hands. They kind of develop like a claw hand over a very long period of time. And same thing with their foot. Their foot kind of will undergo muscle atrophy, resulting in kind of a curvature, call it pes cavus, where the arch of the foot is really high because it's being sort of squeezed up. And so some people have difficulty walking and things like that. It's misdiagnosed as, oh, well, they have arthritis as they get older or something like that. It's like I said, it is getting better now in terms of diagnosis. But yeah, in fact, some of you who are country music fans may have heard Alan Jackson just came out and announced that he actually has Charcot-Marie-Tooth disease. So he's continuing to play for now, but I can imagine that's going to become increasingly difficult for him.
So is this typically an older onset or younger, or is it variable?
So it's very variable, and it depends on the type. So Vivian mentioned type 1A, which is the most common type. That affects about 1 in 5,000 people. So the vast majority of people who have CMT have type 1A. And that usually manifests when kids are little. They may have some difficulty with walking and balance, usually not enough to really capture the attention of most adults, unless they're really looking for it, like, because it is inherited in a dominant way. And the other thing that can happen is they have difficulty running. These kids, because of their lack of balance from proprioceptors largely, that they aren't able to run very well. But then really it's more in the, around the age of 20 when it manifests. There clearly starts to be clear problems in disability.
How is it diagnosed? Is it all symptomatic pretty much?
Well, so usually people will go to their doctor and say, I'm having problems walking, or, I have problems with my balance. And a lot of times the doctor can even, a neurologist can tell just even the way somebody walks. They tend to curve their feet so that the ankles are under a lot of pressure. So many times these people break their ankles very frequently. So then the doctor would refer you to a neurologist who could look for those kind of symptoms. And then the key is they do a measurement of nerve conduction velocity. So they basically put electrodes into two places on your body, usually the leg, and then they'll measure how fast the electric signal goes from one part to the other. And if it's a demyelinating disease, because the myelin is so important for the nerve conduction, then they'll see a difference there in the electrical signaling, the rate that it goes. And then over time, this gets worse and worse. And actually, very frequently is associated with a lot of pain. So a lot of times as people get older, they have a lot of pain.
Are all forms of Charcot-Marie-Tooth disease genetic?
As far as I know, yes. Sometimes it does show up spontaneously. There will be mutations. But I should point out that there's quite a few people that have diagnosis of CMT where we have not identified any gene that's mutated. And there's about 100 genes that have been identified that can lead to different forms of Charcot-Marie-Tooth. Obviously, most of them are quite rare because it causes CMT1A. That's the vast majority. But a lot of people, they still don't know. My guess would be that it's probably a multi-genetic problem where more than one gene is affected, and as a combination, it leads to a similar phenotype.
Is there any evidence, like in MS, there's some people think, or there's possibly some people who it's a combination of their genetic makeup and then an encounter with some sort of pathogen. Do you think there's any evidence that might be coming into play more and more as we start seeing more people who have symptoms and being like, maybe we should pay attention to this and figure out what's behind it? Is there some room for that?
Well, I guess I would hedge a little and say, I think certainly the environment could have a role. And I think that may be contributing to why we see some variability even within type 1A. I have not heard of anything with a pathogen specifically, though. But, it's an interesting idea. So, you mentioned autoimmunity. So some of you may have heard of Guillain-Barré syndrome. This is a syndrome brought on usually by some kind of infection, a viral infection, that for some reason triggers the immune system to target peripheral myelin. Sometimes a viral protein might mimic a myelin protein, although there's really not great evidence for that. But anyway, it basically gets rid of all the peripheral myelin, and these people typically are completely paralyzed. But the beauty of the peripheral nervous system is that it regenerates. So the myelin reforms, and their only risk is, you need to be where you can't breathe. So then they can put you on a respirator, but then virtually everybody recovers from it. So maybe there's something with a viral effect there. And CMT, I don't know. I haven't read anything that indicates any link between a pathogen.
So Guillain-Barré is often called ascending myelitis, right? Where I presume the myelitis goes from foot up. Is there any explanation for that?
Not that I'm aware of, no. I mean, CMT is kind of the same way. So, some of the key diagnostic criteria are it's symmetric, so it's on both sides of the body, and it's distal to proximal. So the things that are affected the most are the distal parts of the nerves. And, we talk about it as a neuropathy because the nerves especially with CMT1A and other myelin-focused mutations. Why should demyelination, or it's really not completely demyelinating, it's myelin isn't formed correctly in most of these forms. Why would the axon degenerate? Obviously, I think to me this gets back to what we talked about earlier, the key signaling between axons and Schwann cells, that if the Schwann cells aren't doing their job and myelinating correctly, then the axon starts to degenerate. And there must be some signaling going on there that causes that.
Yeah, in the central nervous system, I think it's been found that a neuron that was previously myelinated and then demyelinated will die, whereas a neuron that was never myelinated would survive even in that situation. So there's the thought that the axons, the neurons are not, they can survive on their own until they become myelinated. And then it's just like to continue my relationship analogies, it's just like dependency issues, and so then they'll die. So I imagine, that's probably something similar happening there.
Is there, for example, is there any sort of gene expression changes after a neuron has become myelinated? Do you know? Does it sense? Surely it knows that.
How would you propose to do that?
No idea.
Because you can't look. You can't look at the neuron and know that
one by one and get
and know that it's going to be myelinated unless you use a much smaller organism. Because even within some of these nerves that we think of as myelinated nerves, there are a ton that aren't myelinated that are in there. So you would have to. It would be tricky. But it's an interesting question.
I don't know. Perhaps something possible with cell culture. I have no idea. Just right.
I think it's a very interesting question.
Yeah, it's very interesting.
I do want to mention one thing that you mentioned, that non-myelinated neuron. So in the peripheral nervous system, there are a lot of axons that are not myelinated. For example, the ones like if you cut your finger and it still hurts 3 days later, and it's to remind you have this injury and you need to be careful. Those sensory neurons are not myelinated, but they are wrapped by Schwann cells. It's a special kind of Schwann cell called a Remac cell. And that Schwann cell will completely ensheathe multiple unmyelinated axons. It just doesn't form that highly wrapped structure called myelin, but it still ensheathes it. So every axon in the periphery, in the sensory and motor system, is either myelinated or wrapped by those.
So do those get affected by Charcot-Marie-Tooth, the Remark Schwann cells? It
depends on the type. Yeah, it's an interesting question. So in CMT1A, it's a duplication of PMP22. So PMP22 is turned on in myelinating Schwann cells. But I will say there is some evidence that those non-myelinated axons are also affected. Exactly how and why, I don't think we have any idea.
Yeah. I know we have to wrap up here, but did you want to touch on your guys' valiant efforts to figure out what PMP22 is doing and how we can kind of leverage that information, or is that not open for discussion currently?
Well, I would bring up a couple of points there. One is we still don't know what PMP22 does. It's important for myelin. If you get rid of it, you don't get myelin. But exactly molecularly what it's doing, we don't know. It's in the myelin membrane. And there's some evidence it might be important for junction formation, but we really don't know what its function is. And then why does the duplication cause this disease? And again, we have no idea. There's some thoughts about what's happening, but basically we don't know. for some reason, too much, even just a little bit too much PMP22 is a problem. And it's
not clear why that is, because something not me, but people in the lab are currently working on is understanding what kind of stress does having too much PMP22 put on the cell, and it is not obvious.
Right. And I should say that it's not a loss of function, because the heterozygotes have a very different phenotype. It's actually I shouldn't say super. It's a very mild form of neuropathy. It's called hereditary neuropathy with liability to pressure palsy. And the way to think of it is like, how if you cross your legs for a long time, your leg will go to sleep. Well, people who have this, their leg goes to sleep much easier, and then it takes a long time for it to go back to conducting electrical signal.
How long?
I'm not sure exactly. It probably depends on the pressure. If you compress the nerve, then you lose electrical conduction.
Do they, how if you cut off circulation for a long time and then you allow circulation to reflow, you get the worst pins and needles.
Oh, so bad.
So I'm wondering, do these patients have the worst pins of, or is that also blocked? I'm guessing that's also blocked, right? Because that requires nerve to be functioning.
Yeah, that's an interesting question. So I'm not a neurologist. I do basic research, so I have not talked to.
I hope for their sake they do not have the worst case of pins and needles, because that's the worst
my gosh. Pain. Yeah.
But I guess that could probably, like, pins and needles might give, I don't know, but it might give you some feeling of what neuropathy is like. I'm wondering, but it's not pleasant. Really, it's not pleasant.
That's interesting. My wife had sciatic neuropathy, so she had a hemilaminectomy to relieve the bone impinging on the nerve. And the neurosurgeon said it's going to take months for it to spring back before conduction returns. And it did. It did.
After months?
Months, yeah. It took months, yeah. Because she was having sciatic pain. She said, why am I still having this? He said, just be patient. It takes time. I didn't know that. I figured they'd boom. But as you just said, they get compressed, and maybe she.
I don't know. I mean, again, I'm not an MD, but I can imagine that when you have that chronic pain like that, there are also in the sensory ganglia called satellite glia, and they seem to be involved in signaling with neurons when you have chronic activation like that. So it may take a while for those to resolve as well.
So it's neuropathic pain where the initial injury is no longer there, but the signaling pathways are still activated and need time to simmer down.
Yeah, exactly. Again, I don't know that. But I do know that in a lot of chronic pain situations, those satellite glia keep the activation going even after the neuron injuries stop.
But these are different type of glial cells, not Schwann cells.
So actually, they're very, very similar.
Oh no!
The satellite glia look, molecularly just like an immature Schwann cell. In fact, if you take out the ganglia in a early state, like in an embryo, the satellite glia are all in there in the immature state, and then you put the cells in culture, the satellite glia will actually become Schwann cells and myelinate.
Okay, so they're immature Schwann cells.
But basically, nobody knows what's going on.
But they in the adults, they clearly have very different functions. They're not acting like immature Schwann cells. They're buffering neurotransmitters and ion concentrations.
So they're more like astrocytes.
That's really
in that sense.
A lot of people think of them like astrocytes.
Yeah. Mm-hmm. Yeah. So I know we have to wrap it up, but I'll just say something that this is making me think of, which is actually an interest that Bruce and I have. I feel generally like there's so much in the peripheral nervous system that's not known, and it's just not getting as attacked as much by the research community as stuff is happening in the central nervous system. I don't know if that's true, but that's just kind of my feeling, that there are these questions, these fundamental questions, and I'm like, why has nobody looked at that? Like some of the stuff Tim was mentioning, might be difficult to look at, but like what it seems so basic. And one of those is the how do you take the neural crest derived cells, which become satellite glia and Schwann cells. It is unclear what happens between becoming an immature, what they're calling, I guess, or Schwann cell precursor, which at that point it could become a Schwann cell or it could become satellite glia. It's like not necessarily clear what happens at that junction, but it's also totally unclear how a Schwann cell precursor decides that it's going to become a myelinating Schwann cell or a Remax Schwann cell. And if you look at electron micrographs of those two cell types, you'll see that they are morphologically really different. And so there's got to be huge changes that have to occur, but we don't know what leads up to them. And so, for any budding peripheral nervous system scientists out there, start thinking, get those wheels turning, and let's figure out how to answer some of these questions.
All right. That's TWiN number 23. You can find the show notes at microbe.tv/TWiN. Send your questions and comments. TWiN@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Our guest today from Vanderbilt University, Bruce Carter. Thank you so much for joining us today.
Oh, it was great fun. I could talk about the peripheral nervous system all day.
Yeah,
I was like, I should probably keep an eye on time because we could easily talk for another
Sure, no
hour.
problem. We love to talk, and we can just see a tooth there in the background in the tank, right?
That's little
yellow tooth. Tim Chung is at New York University. Thank you, Tim.
Thank you. Thanks, Bruce. Thanks, Vivian. That was very informative. And I look forward to learning a lot more about the peripheral nervous system and Schwann cells when Vivian talks about it in the future.
Yeah, my knowledge is only limited to what herpes simplex virus does, which is to go latent in it, in peripheral ganglia. Vivian Morrison's at Vanderbilt University, or you can find her on LinkedIn. Thanks, Vivian.
Thank you so much. Thanks, Bruce, for coming.
My pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back soon.